Très bien, on va commencer. Je suis ravi d'accueillir Aubrey, que je connais maintenant depuis très longtemps, qui est infectiologue qui travaille à Southampton et qui a beaucoup aidé au développement du vaccin contre le méningocoque B. Et qui est... Are you still editor-in-chief of Journal of Infection?
Oui.
Ah. Et qui est rédacteur-en-chef de Journal of Infection. And
also current opinion in infectious disease.
Ah, good.
Yeah.
Or it is, sorry. So I'm very happy to have him here, and he is very... I was interested, but what he is doing, because we were once, a long time ago, we were invited to make a kind of press review of infectious disease in a congress. Remember this?
Yes.
And I think it was really interesting, the wideness of his knowledge in reporting this in infectious disease. So it's very rare that people get a very wide view of infectious disease. Probably this is a reason why he's continuously interested in staying editor-in-chief of a general journal that gives you this overview of things, of everything in the same time. So you get a very large view of infectious disease in general. And I think it was really interesting to discuss with him, and then since then we're friends together.
Thank you, Didier. Merci beaucoup. Also, I cannot trust any other UK ID academics to keep the impact factor above 5, which is hard. Anyway, bonjour, mesdames, messieurs. Heureusement, je parle anglais. So I'm sorry. You would not like me to speak French. It would be very bad. Now, am I understanding that some of you are PhD students, and you're PhD students and medical students? De médecins, étudiants de médecins, oui, là-bas. And others are scientists. So a real mixture. Well, thank you. So, I'm going to talk about Neisseria meningitidis. Does anyone work here on Neisseria meningitidis? Is there? Okay. So I will thank you for telling me that. So because I will go slowly through the first part of the talk then, and this is Neisseria meningitidis. You currently have a problem in France with this disease, if I'm not mistaken. I don't know what it's like in Marseille, but I know that certainly in parts of northern France there are problems with meningococcal disease. And the organism is a gram-negative Diplococcus, and this is a picture of the major antigenic components of the organism. So it has a number of interesting features. The first thing is that very famously it has a capsule, which is a polysialic acid capsule. And of course, all doctors know about this capsule because that is the basis of serogrouping. So the nature of this capsule as a major immunogenic antigen is the way that we epidemiologically type this organism. But there are other things that are very interesting about this organism. The first thing, that it has a number of interesting outer membrane components, such as pore A. And this protein is huge. It's a trimer. It's a trimeric protein. It is massive. Its function is to allow the passage of electrolytes. And it is very immunogenic, so it generates a strong immune response. There is, like other organisms, certainly like other gram-negatives, it has pyelin, and it uses this to adhere to surfaces. But then it has some other interesting things, such as factor H binding protein. This protein allows it to capture factor H, and that enables it to deter the effect of complement and stabilization of the terminal membrane attack complex. If you look at the electron micrograph, another important feature of Neisseria, like other gram-negatives, is that they make those little, do you see there are some vesicles coming out? So they create these little vesicles that are based, they're outpouchings of the outer membrane, and they contain the cytosol of bacteria, and in the membrane of the vesicle are expressed all of the outer membrane proteins that the organism has. So these are called outer membrane vesicles. The abbreviation is OMV. So when I say OMV, I'm referring to those small products. Okay, so now please can you ask questions if there's something you want to clarify. It's best to say it during the talk than at the end. So please don't be shy. I'm very I like spontaneity. But do you want me to speak for 45 minutes or?
Yeah.
Okay. Right. So during the 1930s, the literature exploded because there was a lot of American outbreak disease, there had been a lot of French outbreak disease, and people started to study Neisseria meningitidis. But at that time, they only had agglutination as a technique of typing the organism, and so that was the primitive form of serogrouping that we use today. Nowadays, we know a lot more about this organism based upon genome sequencing and the use of multi-locus sequence typing, which in the case of Neisseria uses 7 housekeeping genes, and the sequence variation between those housekeeping genes determines the sequence type. But genome sequencing has allowed us to see that this organism is simply a cousin and a very recent cousin of the commensals of the oropharynx, such as Neisseria lactamica and Neisseria polysaccharia. Now, these organisms are adapted to just colonize your nasopharynx. And they do that very successfully. They do not cause disease. And then from these commensals, a branch came off of Neisseria gonorrhea. Now, the distinguishing characteristics of these organisms is that they do not have that polysaccharide capsule. Neisseria meningitidis emerged probably from an early branch of Neisseria gonorrhea, and its distinguishing feature was that it acquired a stable capsule, a stable polysaccharide capsule. So this gave this organism the potential to invade the bloodstream. All right? Now, gonorrhea does have the potential to invade the bloodstream, and probably that's by sialylation of its terminal sugars on its lipo-oligosaccharide. But it does not have a polysaccharide capsule. Likewise, we have recently seen disease caused by these commensals in patients who are receiving certain biologic therapies that inhibit terminal complement. But under normal circumstances, this organism, that organism cannot cause disease. This organism causes disease by invading after a stable period of colonization. This organism is adapted to transmit sexually and to utilize the genital tract. Okay, so most doctors are familiar with this form of epidemiology. So based upon these serogroups. Now, as you can see, in red is serogroup A, orange is B, C is grey, W is a sort of magenta type colour. And this is the traditional way of typing Neisseria meningitidis. And as you can see, the world is different. In some parts of the world, for example, Africa, we have traditionally seen a lot of serogroup A disease. Currently in Europe, we see mostly serogroup B, and this is true in France. Most of your disease in France is caused by serogroup B, certainly until recently. In the United States, they have a sort of equal distribution of B and A and C. So these are all the serogroups that we see distributed around the world. Now, the important thing about this is that we have vaccines which can protect against these serogroups. So vaccines that use the capsular polysaccharide. The thing that you need to know about this is that this capsule, B, is not very immunogenic. Okay, so whereas we can take the C capsule, conjugate it to a protein, and generate high antibody titers, if we try to do the same with B, we cannot do it. The other ones can all be used as vaccines. So it's important to know about this epidemiology to devise your vaccine strategy. Okay, but recently we've learned that the story runs much deeper than serogroups. There is a biology going on that we need to understand because there have been certain events recently that have been very surprising. Now, using multi-locus sequence typing, we can break down the clones of Neisseria meningitidis into a large number of sequence types. Of these sequence types, there are 4, I'm sorry about this particular slide, there are 4 that are associated with disease. And these are shown here. There's particularly, in France, you have a lot of this clone, 269, but a big problem emerging both in my country and in your country is this complex, clonal complex 11, and also 23 and 44. So these are clones of Neisseria meningitidis that spread between our population, and sometimes they emerge explosively, and sometimes they wane and wax. So it's quite interesting. So what these two graphs show here, shows the age distribution. This is in the UK, where we did MLST typing of a huge number of samples from the bloodstream of patients. And this shows different ages. You can see that different ages seem to be infected with different clonal complexes. And likewise, if we look at year on year, so this starts at sort of 73 and now it works up to 2016, you can see that there is a change in which clones are succeeding in the country. And you can see that this yellow one came to a big stop in 1999, and that was clonal complex 11 in the UK. Can anybody think why that might be? Why would a clone stop? Why would a clone stop being? Well, the answer is we vaccinated our entire population with a vaccine that hit a serogroup being expressed by this particular clone. This is European data. It's quite old, but along the x-axis here, these are the most prevalent clones in Europe. Okay, or they were 10 years ago. It's a little bit different now, not very different. And then these colours here represent the different serogroup, the capsular polysaccharides that these organisms have. And the first thing to notice is that some of these clones are more common than others. So, for example, across Europe, sequence type 44 and sequence type 11 are very common. The other thing you will see is that they do not all have the same color. So clones can express one or more different capsular polysaccharides. Very interesting, but from a doctor's point of view, it's very important information, because if a clone can expand, but it can express different serogroups, then you maybe cannot base your vaccine strategy on the clonal biology that's going on. So you need to understand exactly what these clones can do. Something else very interesting, you see those little red lines up there, is that the clones are slightly different, well, very different actually, in the disease that they cause. So for example, this clone here, clonal complex 11, has a much higher fatality rate than other clonal complexes. And I have to tell you that we do not understand why that is. Okay. So I put a picture of in français it's a loup, oui, loup. So in English we have a phrase that says a sheep in wolf's clothing. Does that make sense? En français, sheep in French. Well, this organism is a wolf in wolf's clothing. Okay, it's a vicious clone, and it has expressed different serogroups at different times. So it caused serious outbreaks of serogroup B disease and serogroup C disease in Europe and the United States in the 1960s. It was responsible for a massive surge of serogroup C disease in the 1990s. And actually, it's the reason why I started to do research on this disease. It was because I was looking after 18-year-old children, young adults, with very severe meningococcal disease in the 90s, and they were dying in front of me, before I could get them to the intensive care unit. And we had a big problem, and it was this clone that caused it. It was responsible for the expansion of serogroup W disease that we associated with the Hajj. The Hajj is a mass, it's a mass gathering in Saudi Arabia, Mecca. Many, many thousands of people come together and congregate to worship, and they come from all over the world. And of course, that has given the opportunity for this organism for clones to expand within human populations. Remember, this organism is a human-adapted organism. It doesn't have an animal host that we know of, but mass gatherings is definitely a risk factor for it. More recently, this clone caused outbreaks of disease in men who have sex with men, and also there has been a very interesting expansion of this clone in South America, and that clone has now penetrated European populations, and you're seeing this in France, and I'll show you the data very shortly, because various sublineages of this clone have started to emerge in Europe now, and some of them cause exceptionally severe disease. So this is France, and you can see this shows at the top there the situation in 2010, and here we are referring, I said that most of your disease in France is due to serogroup B. Well, you over recent years, like us, have seen more serogroup W. The relevance of this, of course, is that you can easily protect against serogroup W with a new quadrivalent vaccine, but you cannot easily protect against serogroup B. So this just shows an expansion in France over the last 10 years of a serogroup W clone that has emerged out of several sublineages. Some of them you've imported from the UK, some of them you've imported directly from South America, and some of them have come into the UK from South America and expanded. You can see that the thing that's happened par excellence is that there's more yellow. And that yellow is our UK strain. And our UK strain originated from a South American clone. So this just shows how a clone can enter a population and can succeed and replace other clones in terms of causing disease. Okay. I'm now going to show you some recent data, not from my group, but from Dominique Cogin, who works in Oslo now. And it just illustrates what this organism is capable of, because it is a panmitic organism, which means that its DNA is so transformable that it can change its nature in a very short space of time. And so around about 5 or 6 years ago, in Niger, it started to become apparent that there was a very severe outbreak. And before then, most disease in Niger had been either serogroup A, although that was being brought under control with a vaccine program, or it was quite a lot of emerging serogroup W. But then all of a sudden, there was a very severe outbreak of serogroup C disease, which was really unexpected. And then it went into Nigeria, and then back into Niger, and it gained momentum. It became a very severe disease that these people were experiencing. And Dominique Cogin's group basically acquired these isolates, and it was assumed that it would be a clonal complex 11 strain that had acquired a C capsule, but it wasn't. It was a bizarre variant of a novel sequence type called 16188. And this was unexpected. And over towards the right there on this distance map, you could see that there's a Burkina Faso carrier. So this isolate turned out to be a strain that had been known about in Burkina Faso, but there it was associated just with asymptomatic carriage. It was never associated with disease. And yet it was very close to the strain that was causing so much disease in Niger. So this was then investigated, and what had happened was that this carrier strain, which was not capsulate, it had an ungroupable capsule, actually acquired en bloc an entire capsular operon, all of the components that you need to make a capsule. And it was capable of expressing serogroup C. But it also acquired a prophage, which was capable of generating, was associated in other sequence types of Neisseria with very severe disease. So somehow you have the situation where an organism in carriers in Burkina Faso has acquired capsule and then causes, because it's also got a prophage in it, causes a massive expansion of severe invasive disease in Niger. This was eventually brought under control, and I'll come back to that. Something that is of concern, coming back to Europe, coming back to France, coming back to the UK, is that our clonal complex 11, which currently is either carrying W or C, could start to express serogroup B. And in fact, we are just starting to see this now, certainly in the UK, and you have also seen it now in France, that this wolf is starting to make serogroup B And as I said to you, the vaccine against serogroup B is less reliable, and I'll go into why that is. Okay. Any questions so far?
Did you look at CRISPR?
Sorry?
Did you look into emerging clone? Are they CRISPRs in
well, it wasn't my work, but I didn't see that. I didn't see it in their list of genes that were. So I don't know if they had a CRISPR system.
Because one of the things that determines that the clone is occurring that when you lost CRISPR, then you can be colonized
by anything. Yeah. Well, that's interesting. I didn't see that, but it wasn't. It was Dominique Fougin's work, so but they didn't comment on that.
Well, I think that not a lot of people realize that usually an emerging clone starts by losing the capability to clear out foreign DNA.
Yeah. Any questions? Okay, so now I'm going to talk about vaccines because it's relevant to what I've been talking about before. Now, the history of meningococcal vaccine is that there were some primitive vaccines. These just took the purified polysaccharide and inoculated people, but these vaccines were really weak. They didn't engage T cells, there was no memory, very short-lived antibody production, months, not very potent. But then, like with the pneumococcal vaccine, about 20 years ago, we started to use glycoconjugate vaccines where they were conjugated, the polysaccharide was conjugated with a protein, usually either a toxoided derivative from the tetanus toxin or from the diphtheria toxin. And these generate a very strong immune response with good memory and high titers of antibody, and they use T cells to do that, and are very, very effective vaccines. And there's now a plethora of these vaccines. The most recent of which is the pentavalent vaccine, which contains A, C, W, Y, and X. But you'll have heard of the quadrivalent vaccines. I think you're starting to use these in France. Or you were talking about deploying the quadrivalent vaccine in France. I don't know if it's happened yet, but these are the conjugate vaccines. This just shows you an effect. I told you that I was a junior doctor in the UK and I was seeing patients with C disease. Well, during the late 90s, we were seeing in the UK 3,000 cases a year, and half of these were in teenagers, and they were exceptionally severe, and our death rate was like 20%, which is really vicious and very nasty indeed. But what we did was, before we had even done the phase 3 clinical trials for this vaccine, we basically gave the whole population up to the age of 18 the glycoconjugate C vaccine, and that's what happened. And that's been a tremendous, tremendous success. Another big success, however, and even more spectacular, was the deployment in the meningitis belt of Africa of a conjugated serogroup A vaccine, or Menafrivac. This was a paper published in Lancet showing the result of giving just in one country this conjugated serogroup A vaccine, and this is Chad. And what the team did was that they had populations that they could vaccinate and others that they couldn't, and they looked at the limited surveillance data they have, and in blue there you can see what happens to populations that did not receive this vaccine, and in red is shown the population who did, and the attack rate per 100,000. And that's, look, weekly incidence. So 5 per week, that's 250 per 100,000. That's a huge attack rate of this disease. But the disease was obliterated with Menafrivac. And when they finally completed the rollout, the entire country, and all those countries that were part of that program, have seen a dramatic reduction in serogroup A disease. So I told you that serogroup B is difficult to protect against. That's because the polysaccharide is not particularly immunogenic. And the reason for that is probably because it's very similar to a host protein called neural cell adhesion molecule 1. But there are now 2 vaccines, one of which is Bexsero and the other is Trumenba. Sorry, that's a spelling error. They're quite different, but a bit similar. Bexsero, which was developed by Rino Rapulli and his group, including Maurizio Grazia Pizza, contains 4 components. So it contains, I told you about factor H binding protein. It also contains NAD-A, or Neisseria adhesion antigen, NHBA, but it also contains an OMV, an outer membrane vesicle, which contains a pore A protein. And the vesicle that they use is the same one that the New Zealanders tried to use during the early 2000s to try and control a nasty outbreak of B disease. So it's got an OMV with pore A in it. It's got 3 other antigens. It's been licensed. We've started using it in the UK. The other vaccine is Trumenba. Now this just uses 2 subvariants of factor H binding protein. So it relies on that protein to generate an immune response. Both of them require at least 2 doses, and both of them are jolly painful. I have personally given this vaccine to 1,000 people, and I can tell you that it hurt them. So anyway, so going to Bexsero, the first of those 2 vaccines, the big problem with this vaccine, all right, and why it's not the perfect solution, is because the abundance of these proteins in the population is not 100%. In fact, this just shows the UK proportion of our isolates that express these antigens. We have none of the NAD-A in our UK isolates. And I think the same is true of France. We have very little immune response to the pore A that's been used. And we rely very much on NHBA or factor H binding protein, and even they are not universal. So as vaccines go, it's not the best. Okay? Let's be absolutely clear about this. I sit on the joint the committee in the UK which deploys vaccines to our children, and this is the situation we were looking at in 2015. I have to say, most of us on the committee had lived through this period, and we were sick and tired of serogroup B disease and not being able to do something about it. The truth is that actually you can see in blue there, in blue is serogroup B. This is the UK prevalence of these organisms causing invasive disease, serogroup B. Serogroup B is our commonest serogroup, but you can see if you look at this graph that actually things are changing over time because clones emerge. And so B has actually been going away. And that's actually true of this country, and it's true of other parts of the world. And then the yellow has started to expand, and that's serogroup W, which is happening in France, as I just showed you. Here is our serogroup W expansion. This shows the monthly isolates, and the one in red is the most recent when the data was collated, and an expansion of W. And not only is it expanding, but it's tending to infect different populations. So meningococcal disease is changing. It's now a disease that we're seeing as often in adults as we see it in children. And that's because of the emergence of this clone, which for some reason is infecting adults. More common in adults than in children. That's strange. We responded to this in 2015 by giving the quadrivalent vaccine that contains W, and then subsequently in the solid blue line you can see that we've started to see some deletion of our W disease, something that you can do in France. But going back to serogroup B, we made a decision in 2015 that we would give every single infant in the UK Bexsero. All right? And we listened to our modelers and our health economists, and they said, yes, it is. Yes, it is cost effective, because before we can deploy a vaccine, it has to be cost effective. And I have to say, the mathematics was tortuous. But we deployed the vaccine. And we gave it to our infants. We did not give it to our adolescents. And this is important because most of the carriage of Neisseria is in adolescents. So this just shows what happened to serogroup B disease over 5 years in ineligible children up to the point at which we were deploying the serogroup B vaccine. The disease was actually going away. So it's a big challenge to measure the effect of a vaccine when the disease is disappearing. And the red spot that you see there was 6 months after deployment of the vaccine, and you can see that there was a lower than expected incidence of disease in that period compared to expected. This was computed to show a vaccine efficacy of about 80%, which is quite good for a vaccine. So according to the statisticians within Public Health England, it worked. But in Quebec, they also did the same thing, but in the case of Quebec, they vaccinated people up to the age of 20. And they also had an effect of 80%. So that finding has now been reproduced in 2 territories. So this vaccine probably can work in protecting the vaccinated population. But if we get an expansion of serogroup B disease in clonal complex 11, that could be a different matter. The disease, the serogroup B disease I've been talking about, actually is mostly a clonal complex called 269 and some clonal complex 44. If we started to see an expansion of this clone expressing serogroup B, we'd be in deep trouble. And that is because our current serogroup B expressing clonal complex 11 only has one component of the Bexsero vaccine. And what that means is that there is every chance that there will be escape from the vaccine. There will be vaccine escape. And then that will be a big problem. Here's something very, very, very interesting. I showed you the distance map earlier where Neisseria gonorrhea was very close to Neisseria meningitidis. In fact, meningitidis branched off gonorrhea probably about 400 years ago. So, and you can obviously do that by computing the SNP frequency. But it was noticed that gonococcal disease had become less common in certain places where OMV vaccines had been deployed. So, for example, the OMV vaccines were deployed in the 80s in Norway, in the 90s in Cuba, and then the New Zealanders gave an OMV vaccine. So this is a very simple vaccine to make. You just spin down some meningococci in an ultracentrifuge, you collect a layer that's got outer membrane vesicles in it, and you inject it into people. It stings, but it's got the components of that organism. So the New Zealanders in the early 2000s had a big problem of serogroup B disease in their Maori population. They gave them all OMV, eventually, it took them a long time to do it, but they gave them all this OMV, this New Zealand vaccine generated in OMV. And what they were able to do, because they had quite a lot of gonococcal disease, was to track the incidence of gonococcal disease and then do a case control study on the people who had or had not been exposed to this OMV vaccine. And what they saw was a significant impact of gonococcal disease in cases versus controls, cases being people who've received the vaccine. The red line there shows the incidence of gonococcal disease. And the blue bars is the incidence in vaccinees, and the green bars is the incidence of gonococcal disease in non-vaccinees, in controls. And it appears to be the case that this vaccine protected against gonorrhea. Now, if that is true, then this would represent the first evidence of a successful vaccine against gonococcal disease. Now remember, gonococcal disease is very different to meningococcal disease. Meningococcal disease is a bloodstream disease. It's a disease of the endothelium. Gonococcal disease is an inflammatory disease of the urethra. Very rarely is bacteremic. Different immunology, different vaccinology. Here we have a vaccine that seems to prevent gonococcal disease. Obviously, you can imagine that the entire world is interested in this because it's the first vaccine for gonorrhea. And it opens up the possibility that we might be able to generate a decent vaccine for gonococcal disease using OMVs from appropriate clones of Neisseria. For example, the Bexsero vaccine has the same OMV component. Okay, so now I'm going to go into some science because the dogma has been that the way to protect against meningococcal disease is to give people high antibodies in their bloodstream. And we measure that functionally by saying that they have, by measuring serum bactericidal activity. So the ability of the blood to kill the bug. All right, so we take your blood after we give you the vaccine, and we take a selection of strains, and we see how bactericidal it is. That is thought to be a surrogate marker of protection. In fact, the dogma states that in order for a vaccine to be considered efficacious, that it has to have an SBA titer of 4 or greater. And because of this dogma, many promising vaccines have actually fallen by the wayside because they didn't do this. They were logical, theoretically, but they didn't generate serum bactericidal antibodies. The truth is that the community is changing in the way that we think about meningococcal disease. We now understand that this phenomenon, carrying, is the key to controlling meningococcal disease, because if you do not carry the organism in the oropharynx, you cannot get disease. It is the first step in meningococcal disease. And of course, that is true because we transmit the organism very readily by close social contact. And it's that transmission process that is the key to controlling a disease like this, the disease of a pathobiont of the oropharynx. Who carries Neisseria? it's students. If you look at this, this is the prevalence of carriage, the prevalence of carriage. This is age, 20-year-olds at the peak. So you guys, you're carrying Neisseria meningitidis, okay? So, you know, young people carry Neisseria meningitidis. Bizarrely, young infants don't really have a high carriage rate. And students do it because they smoke, they live in dorms, they go to nightclubs, they kiss, etc. And the other people who get it are, of course, who acquire the organisms are household contacts of index cases. But the key thing is this: if you can't carry the disease, you can't transmit it. And if you don't transmit it, your population doesn't have an expansion of this disease. So the carriage rate in here, 1 in 10 of us in this room is probably carrying Neisseria meningitidis. You guys up there, it's around about 1 in 3 of you, 1 in 4 maybe. The guys down the front, it's all 1 in not carrying Neisseria meningitidis. So, you know, disease though, really interestingly, your chances of getting disease if you're a carrier are really low. Now this is very interesting. Very low indeed, because if you're all carrying now, let's say 3 of you are carrying a serogroup W, clonal complex 11, in your oropharynx. It's possible. This organism is in France, so it's possible you're carrying it. I can tell you that your chances of getting meningococcal disease are 1 in 10,000, if you're a carrier. If you're not a carrier, it's 0. So, now, this is quite an interesting graph. This shows the disease rates of meningococcal disease, and this, in the dotted line, I talked about the serum bactericidal antibody. If you've got high SBAs, you do not get invasive meningococcal disease, okay? At least 120, you have a 0 chance. Over 4, you have a very small chance of getting meningococcal disease. But only 50% of the vulnerable population actually have high SBAs. And I've told you that the force of infection is like 1 in 10,000, even for the most virulent clonal complexes. So there's something else happening that's got nothing to do with SBAs. And so the dogma is wrong. All right? You do not, SBAs are not the limiting factor in disease. They will stop you getting disease, but something else can stop you getting disease. When I showed you the graph showing that we obliterated C disease from the population, we used a conjugate vaccine. We subsequently learned that in the population that we vaccinated, youngsters, that actually the antibody titers, the SBAs, are very short-lived. And yet we saw complete obliteration of the disease from the population. All right? So despite the fact that they were not having antibody around, the disease went away. And it was Martin Maiden who discovered why. Because he did a big carriage study in the UK in adolescents, 1 year after we deployed the C vaccine. And he measured carriage of serogroup Y, of serogroup W, of serogroup B, and serogroup C. And after we vaccinated people with the C glycoconjugate, we saw no change in carriage of B, no change in carriage of Y, no change in carriage of W, but there was a significant drop in carriage of serogroup C. So what this vaccine did, this was 1 year after, all right, it was in a population that had not been covered very well with the vaccine, but was covered around about 60% or so. So what actually happened was that by changing carriage with this vaccine in the adolescent population, we removed C disease from the population. And this is the changing concept of meningococcal disease. Now, what's shown there in the green is what the modelers would have thought would have happened to C disease in the absence of this effect on carriage. And in blue is what actually happened. So clearly, this impact on carriage was highly significant. Now, going back to Chad, what they did, I showed you the obliteration of disease there in red, but they also found a reduction in carriage. And subsequently, that disease, that serogroup A disease, has remained very low in Chad because of this impact on carriage. Very quickly, I measured this for serogroup B. And what we found was that this Bexsero vaccine has whereas this shows the carriage rate, the effect on carriage of serogroups C, W, and Y after we gave a quadrivalent vaccine. As expected, a big significant impact on carriage of serogroup Y. When we looked at the same thing with Bexsero, we saw a much weaker effect. And in fact, we saw very little impact on serogroup B carriage. So, the big difference between Bexsero, Trumemba, and the quadrivalent vaccine, the conjugate vaccines, is this. We can expect, like a conjugate vaccines, to generate a sustained protection, herd protection. We cannot expect the same thing with these protein-based vaccines, even if we give it to adolescents. All right, so it's not going to happen. Now, I think I'd better stop there because I have come to the end. But there is one thing I want to show you, though, before I do, which is this. A brilliant paper from Médecins Sans Frontières, what's it called? Epicentre. Is it? Epicentre? Where they went into, I told you about the serogroup C outbreak in Niger. They investigated the use of chemoprophylaxis. Now, what that is basically giving people ciprofloxacin, it's a very hard thing to do because the environment is difficult. It's very difficult to administer such a program. But what they showed was that when they gave ciprofloxacin, there was a strong impact on the incidence of meningococcal disease. Interestingly, if they saw a case of meningococcal disease and they gave Cipro to the whole village, there was a dramatic reduction on disease. If they did just household prophylaxis, which is what we do, and what you do in France, I think, they saw no impact on the overall incidence of disease. I commend this study because it was incredibly difficult to do this. And it was done by a guy called Calderon and Rebecca Grace, who work for Epicentre in Paris. So I'm going to stop there, and thank you very much. And I'm sorry I've just gone slightly over time.